Hey everyone, welcome back to my class. In today's section, we are going to study about the flagellar and ciliary movement. In the previous video, we have already seen about the structure of flagella as well as the structure of cilia. Both are same, correct? In today's class, we are going to study how cilia and flagella are useful for the locomotion or for the movement. Let's begin our class. Now, I want all of you to prepare this chart so that it will be easy for you to remember the concepts. Okay, everything has to be there in one page. Flagellar or ciliary movement. When we talk about flagellar or ciliary movement, there are four types of locomotion we are going to talk about. What are the four types of locomotion? One is your swimming locomotion, second one is your flagellar locomotion, third one is your ciliary locomotion, and fourth one is your gliding locomotion. Now, if we talk about the swimming locomotion, under swimming locomotion, we are going to study about the bending movement of flagella and cilia. Under flagellar locomotion, we are going to study about the undulation movement, sidewise lash movements, simple conical gyration movement. Now, under sidewise lash movement, I want you to mention this as well. We are going to study about the effective stroke and recovery stroke. Okay. Now, the third one is your ciliary locomotion. Under ciliary locomotion, again we are going to study about the effective stroke as well as the recovery stroke. But we have taken a special example, that is your paramecium. Under paramecium, we are going to study about the synchronous movement as well as the metachronous movement. Right? Now, the last one is your gliding locomotion. Basically, it is with the help of myonetes. Right? So I want all of you to write this in a single page so that it will be easy for you to remember. Now let's begin our class with the swimming locomotion. When we talk about the swimming locomotion, we'll be considering this under the in the protozoa. Now the whole chapter is about the locomotion in protozoa, so we have taken an example of protozoa. Now swimming locomotion is brought by both, that is flagella and cilia. According to Hyman, flagella and cilia both are called undulipodia. According to Hyman, both flagella and cilia are called as undulipodia. So whenever I use this word undulipodia, you're supposed to remember that it is another name for flagella and cilia, right? They are for the locomotion in water and body fluid. Now if I'll ask you to go and swim, where are you going to swim? Obviously you will swim in the water. Of course, few exceptions are there. For example, in my class, Hemang is there. Hemang will not swim in water. Hemang is going to swim on land, so that he will, simultaneously he's going to sweep the floor, right? So sweeping work will also be done and swimming work also be done, right? Jokes apart, class. They are for the locomotion in water. And what about the body fluid? Body fluid, when we'll talk about ourselves, body fluid can be your blood, can be your lymph, can be your tissue fluid, or your interstitial fluid, right? So what are parasites? Parasites are the one who feeds on the living organisms. So these living organisms are going to move, will show their locomotion by using their flagella and cilia. This type of locomotion we call it as swimming locomotion, which will happen in the water or in the body fluid, correct? Now, under swimming locomotion, we are going to study about bending movement of flagella and cilia. How it occurs, let's just see. Bending movement of flagellum is caused by sliding of microtubule past each other due to functioning of dynein arms and utilizing ATP. Now, please understand over here, just focus. Let me draw. This is your A and this is your A microtubule. Then we have B microtubule, and here you have B microtubule. Okay, now see here, I'm going to draw the dynein arms. Pair of dynein arms are there, right? Now this dynein arm, this is one doublet and this one is second doublet. Likewise, we have 9 doublets, right? Now this dynein arm is going to attach to the binding site of the other doublet. Let's consider that the binding site is present on the B. Binding site is present on the B. So this dynein arm is going to come and bind over here. So how it will be? Let's see the structure. So suppose this is your microtubule A again and this is A again, and what was the color? B over here, B over here, and now this dynein arm is going to attach to B. Okay, let me join only one so that it will be easy for me to understand. Okay, now but both will be able to join. Okay, let's consider only one. So this dynein arm has joined B. Now what it will do? It will pull this microtubule towards itself. It will pull this microtubule doublets. It will pull doublets towards itself so that there will be the sliding movement. Now see very carefully. Now what will happen? A is over here. Now due to pulling, what will happen? A will come nearby. A will come nearby, right? Let me draw it with same color. B is over here. B is over here. Now, then dynein arm is going to detach from that particular place and it will attach somewhere else. Okay, it will attach somewhere else. Again, pulling action will be there. So, what will happen? This will move, this doublet will move this side and first doublet will move this side. So, what is this? This movement and this movement. What is happening? It is called as sliding movement. So, your dynein arm will attach, slide, detach, and then attach again, then slide again, then detach, then attach again, then slide away. So due to this, what will happen? There will be the movement of sliding movement. There you will see sliding movements among the doublets, and this helps in the bending locomotion. Now sliding movement is the okay, but ma'am, how does this bending locomotion occur? So class, don't forget that these doublets are connected with the radial spoke to the inner sheet, right? So sliding, limited amount of sliding will be there, right? So after that point it won't slide. So because of this radial spoke, what will happen? You will see bending over here. You won't see the straight sliding. You will see bending over here because something is there which is holding sliding, right? What is that something? That is nothing but your radial spoke, correct? Now, let's understand again. Now, bending movement of flagella is caused by sliding of microtubule. I explained you over there due to dynein arms. Now, how that happened, I will be explaining you that. I mean, I have already explained you the mechanism, but what is the role of ATP over here? Now, see class, I told you that the dynein arms are going to attach, detach, attach, detach, attach, detach. For this attachment, what is required? That is some activity is going on, so ATP is required. Can I say that only by the presence of ATP you will be getting the energy? No, obviously not. So what you have to do, you have to break this ATP. ATP means we have 4 adenosine triphosphate, right? Triphosphate. So whenever this bond is being broken down, there will be the release of energy. So this attachment, detachment is because of this release of energy. But how this ATP will be broken down? How the bond of phosphate will be broken down? It will broken down by enzyme ATPase. Whenever you see A's, it means I'm talking about the enzyme. So this adenosine triphosphate bond will be broken down by enzyme ATPase, which utilizes water. So in the presence of water, ATPase are going to break the bond from ATP, and this releases the energy for attachment and detachment of dynein, which cause the sliding movement of doublet. Right? I hope this is clear to you. Now this dynein arms are going to pull the neighboring doublets, correct? Let's come back over here. A flagellum pushes fluid medium at right angle. This we have already seen in yesterday's class. Suppose the movement of water is in this direction. So the movement of flagella will be always perpendicular to the direction of water. So flagellum pushes the fluid. Now flagella is going to push the fluid like this. So when it is pushing the fluid like this, it is going to be at the right angle from the point of its attachment by its bending. So this bending which is happening, suppose this is there, this bending which is happening, it happens at the 90 degree, correct? Now, let's see the mechanism. Dynein arm of each doublet attached to the adjacent doublet. That's what I told you. One doublet is there, then we have another doublet, and binding site will be there. There the dynein arm will go and bind, right? Which pulls the neighboring doublet. So this neighboring doublet will be pulled. The doublets slide pass over each other. Due to pulling, what will happen? This doublet will slide over this doublet, right? So it passed over each other. But how it is? The first one will go in this direction and second one will go in this direction. direction. So it is the sliding in opposite direction, right? The dyning arm release and reattach. So once that pulling is done, again dyning arm will detach and attach to another binding place. It will attach, binding point it will attach, and this will help in further sliding movement, right? So see here, it will reattach a little farther on the adjacent doublet and pull again. The doublet of flagellum and cilia are physically held by radial spoke. How is radial spoke? It is elastic. So it will allow, due to its elastic nature, what will happen? It will allow little movement. It will allow only limited amount of sliding, right? The doublet cannot slide past much because of the presence of radial spoke. Instead, they curve. So since there is limit, they won't slide like this. There will be a curve because something is holding. What is that something? That is your radial spoke, right? Now, such bending movement play an important role in flagellar and ciliary locomotion. Correct? Now, if Nexon and radial spoke, so Nexon is the one which is going to connect the two doublets, okay, and radial spoke is connected with the inner sheet. So if both are subjected to enzymatic digestion, now if I'm going to use enzyme and this radial spoke and nexines are broken down, what will happen to the doublets? Will you be able to see the sliding movement? No, there won't be any sliding movement. Instead, doublets will be moving here and there. Let's see this picture given in your textbook. So this is the microtubule doublet, and these doublets are going to bind with the dyning arm, not bind with it but attached to the dyning arm, okay. Now this dyning arms are going to see, dyning arm head has ATPase activity, okay. So this is going to utilize the ATP and it will help in the bending movement. Now suppose if I talk about like this, so whenever there is a bending, so obviously one arm will be larger as compared to the another arm, right? So that's what's all about the bending movement. Let's jump on the another locomotion, that is your flagellar locomotion. Now flagella shows three types of locomotion which we have already seen, that is undulation movement, sidewise lash movement, simple conical gyration. Now one by one we are going to see all the movements. First one will go about the undulation movement. We have already discussed about undulation. What is the meaning of undulation? Undulation is nothing but the wave-like movement. The meaning of undulation is nothing but the wave-like movement. Here you can see how your flagella is moving. It is giving the wave-like appearance, right? Now, undulation, two types, actually three types you will see, but let me consider the two types. You will see from base to tip. The wave-like movement, either you will see from base to tip or you will see from tip to base. Based on that, different things will be present. Let's discuss that. So if there is undulation from base to tip, so if there is wave movement from base to tip, now this is your base. Suppose consider this by to yourself. Let me consider this as a Gia. Okay, so this is Gia. Now Gia, here is the base and here is the tip. Tip is always far away, right? So tip is far away from Gia. Now if Gia wants to throw something, or Gia is holding something and she wants to do the wave movement with that. So obviously from base to tip, if I want, that means what I have to do? I have to push that something, right? I have to push that something. So whenever you see whenever you will see wave movement from base to tip, what will happen? You're pushing away somebody, right? Base to tip, away, that means you have to push, okay. Towards means you have to pull, so away means you have to push. So Gia is going to push. So here basically you will see the pushing force will be there, and when you're pushing someone, that someone is going to come towards you or it will move back. Of course it is going to move back. So same thing will happen over here. Whenever there is a wave movement from base to tip, what will happen? Pushing movement will occur. Due to the pushing movement, what will happen? The organism will move in a backward direction. For example, your propeller of boat, okay, propeller of boat is going to do the pushing force, right? Now the another one, if I want to consider from tip to base. Now this one, another example, suppose this is Danya. Okay, I've taken an example of Danya. Now, Danya wants someone to come near to her. Now, that someone like in a wavy movement. Okay, so what she has to do? She has to pull. Okay, she has to pull. Suppose she has some ball which is far away. Okay, so what? And that ball is linked with a thread. So, if she wants that ball near her, what she has to do? She has to pull the ball, right? So, from tip to base, if you will see the wave movement, that is nothing but it is due to pulling force. Base to tip, away means push. But tip to base, it is pull. Tip to base, it is pull. So when you are pulling someone, that means that someone is coming near to you. So that will be the forward movement will occur. For example, propeller of aeroplane. Whenever propeller of aeroplane rotates, what will happen? There will be the forward movement, right? This was an example given in your textbook. Now see class, until now we have studied two examples, that is from base to tip. One second. From base to tip and from tip to base. Base to tip is nothing but you are pushing away and that is going to be the backward direction. Tip to base, you are pulling and that is going to be the forward direction. These are the keywords you're supposed to remember. Now in both the situation, my flagella was not bent, was not tilted. But what if my flagella is bent towards one side? Here in the picture you can see my flagella is bent towards one side, and if the wavy movement is from base to tip, what will happen? There will be the lateral movement, no forward movement, no backward movement. Whenever flagella is tilted towards one side or bent toward one side, what will happen? The organism will move laterally. Laterally means sidewise movement will occur. Sidewise movement will occur in a opposite direction, right? Now, what is sidewise lash movement? Mostly the movement which you will see in an organism having flagella will have sidewise lash movement. It is the flagellar movement in many organisms. So, this is the mostly you will see this kind of movement. Now, it consists of your effective or propulsive stroke or recovery stroke, right? One by one we are going to study both the stroke. When we say effective stroke, you know, effective English word, if I want to consider, it affects you, right? So something happen over here. But when you talk about the recovery stroke, that means gaining back. Again, you know, coming back in a relaxed mode. For example, suppose a bowler is throwing ball to the cricketer, a batsman, okay? So batsman is in its effective stage and he is going to hit the ball, right? So that is the effective stroke. Now, when the ball is already hit by the batsman, what will happen? Again, batsman is going to come into the relaxed mode, that is called as recovery stroke. Let's see over here what is effective stroke and what is recovery stroke. Effective stroke is nothing but the flagella become rigid, that means hard. But when we talk about the recovery stroke, it is you know, lazy busy. Recovery stroke is lazy busy relaxing state, so it will be soft. जैसे अगर आपका एग्जाम आ रहा है तो you will be, you know, very much tense and you will keep on studying, but when exams are not there, you're relaxed, right? Now, offer more resistant to water. Now it will be having more resistant to water, मतलब water effects are not going to be much on the flagella. It will be resistant to it. Water movement will not affect the movement of flagella, right? But when we talk about the recovery stroke, it is least resistant to water, right? Now, start bending. When we talk about the effective stroke, start bending to one side, beating against the water at right angle. That's what we have studied. See this structure over here. Flagella will show 90 degree, and here the beating movement, you can see the flagella will start beating at 90 degree at its body axis. Flagella pushes the fluid medium at right angle. So at right angle, sorry, when you will see sorry, when you will see this flagella over here, so it is pushing the water at where is it, fluid medium at right angle. Why? Because my flagella is moving at right angle, right? It is moving at the 90 degree. The movement of water is parallel to the body. See, flagella is moving at 90 degree, but what is the movement of organism in a forward direction? What is the movement of water in a forward direction? Isn't it parallel movement? Obviously it is a parallel movement, and organism will move forward. The direction direction of movement is parallel to the body surface. This is the body surface and this is the movement of organism. So both are parallel. But when we talk about the recovery stroke, what will happen? It comes to its normal position. That means move backward to the original position. In effective stroke, the organism is moving in a forward direction. In recovery stroke, it will move in a backward direction. That means comes to the original place. For example, here suppose, suppose the organism is like this. So number 1, number 2, number 3, number 4. You can see this is the effective stroke. Now when we come like this 4, 3, 2, 1 in this way, this is called as recovery stroke. Okay. Now third one, the last one, simple conical gyration. You know the meaning of simple, you know the meaning of conical, that is a cone shape, and gyration. What is the meaning of gyration, class? Gyration is nothing but the circular or spiral movement. It works like a screw. All of you, I think you must have seen the screw. Now when we use screwdriver on the screw, how it rotates? It rotates at its own axis. ऐसा तो नहीं है ना कि यहाँ पे use कर रहे हैं और ये rotate होता होता यहाँ पे पहुँच जाएगा. No, it will rotate on its own axis. For example, if you see the drilling machine will drill at only at one position. How drilling will occur? That machine is fixed with the pointed screw which rotates at its own axis. Due to that, there will be the creation of hole, right? So this gyration is nothing but the circular or the spiral movement. Here flagella turns like a screw. Only at its own position it will rotate like this at its own position, right? This exerts the propelling action that pulls the organism forward through water. Due to this, what happens? See, when I started drilling with screw, only suppose this is the wall. Slowly, here it will be my screw. Slowly, here it will be my screw, right? So what is happening? There is the movement in the screw. Same thing will happen in the flagella. When it keeps rotating, it will start moving forward. That pulls the organism forward through water by a spiral rotation around its axis. Remember, class, at its own axis. It's not going to change the direction, right? And the gyration on its own axis. Both are same, class. Spiral rotation as well as the gyration. Gyration is nothing but the spiral movement. Correct? Here you can see the picture. So your flagella is rotating like this at its own axis. This rotation is nothing but your spiral movement. Here you can see how it is going to move spirally. Don't think it is two flagella. It's just showing the spiral movement over here, right? Now the third one is your ciliary movement. Of course, as the name is ciliary movement, that means we will be able to see this movement in ciliates. It is performed by the bending movement of cilia. Now this bending movement, you remember, we have seen in the swimming locomotion. So this bending movement is shown by flagella as well as the cilia. Flagella may we have seen that bending movement is due to sliding past and due to radial spokes, right? Now let's see the bending movement in cilia. Cilia moves in a water parallel. We have already discussed how is the direction. See here. So if this is an organism, okay, now this is the cilia. If you will see, water is moving in this direction, so cilia will also move in this direction, then it will come back to its original place. So how is the direction of cilia and water? It is parallel. So that's what I have written. It is parallel, which shows pendular movement. That means it will come down, go back, come down, go back. So it will show back and forth motion, right? The movement of water may also be described as perpendicular. Class, see the second picture. When you see the water is moving in this direction, but see this direction of see the axis of cilia. The movement of water may also be described as perpendicular to the axis of cilia. When you see the axis of cilia, cilia is in this direction. See, movement, while movement, the movement is parallel direction. But what is the exact axis of cilia without any movement? That is nothing but perpendicular to the movement of water. Cilia also shows swift back and forth motion like a pendulum, right? These are called effective and recovery stroke, which we have already studied. Now see, cilium bend backward, so पहले वो पीछे जाता है and beat against the water. As the cilia pushes the water backward, see, when it is moving backward, obviously it has to push the water backward, right? The body moves forward. अगर cilia पीछे जा रहा है, तो body आगे आ रहा है. Let me just show it over here. So this is the cilia. If cilia is moving back, body is moving in a forward direction, right? Cilia is moving back, body is moving in a forward direction. In this method, cilia moves like a pendulum. In this method, cilia moves like a pendulum. We call it as effective stroke, right? So here you can see this is 1, 2, 3, 4, 5, 6. This is your effective stroke from 1 to 6. Now then what will happen? The cilium after the effective stroke again come back to its original position by its backward movement without any resistance. So during effective states, it's move forward. It moves forward. During recovery stage, it moves backward. Right? So this was about the effective and recovery stroke. Then the next one is coordinated movement, especially one example if we'll take that is paramecium. Paramecium is going to show two types of movement, that is synchronous movement and metachronous movement. Paramecium is a primitive ciliate which has cilia all over the body, right? Let's see one by one. When we talk about the synchronous movement, it is nothing but it will occur only in the transverse row. We have already discussed about the transverse row as well as the longitudinal row. So this here you can see, this we call it as transverse row. And when we talk about the longitudinal, in this way when we are taking, we call it as longitudinal row, right? Now, so if I talk about the synchronous, it will always occur in the transverse row. It will always occur in the transverse row. Then what about the metachronous? It will always occur in the longitudinal row. It will always occur in a longitudinal row, right? Now, what is actually synchronous? S for synchronous, S for simultaneously. When together all the cilia beat, simultaneously means together, all together, when all the cilia moves in one direction, we call it as synchronous movement. Then what is metachronous movement? When the cilia moves in a sequence wise manner. Here you can see, suppose let's consider this one. So first this one will move, then second this one, third this one, fourth this one, fifth this one. All the cilia are not moving together, one by one in a sequence way. It will move in one direction. This movement passes like a wave. It resembles the movement of plant in the paddy field due to blow. So when you see the paddy field due to blow of the wind, what will happen? You will see it will show the wave-like movement. So what is synchronous? When all the cilia simultaneously beat in one direction in a transverse row. What is metachronous? When all the cilia moves in a sequential way in one direction, that in a longitudinal row, we call it as metachronous. Let's see one example over here. So if you will see the example of trachea, you can see all the cilia are moving in one go, right? Together, all together, simultaneously it is moving. But when you see in fallopian tube, one by one the cilia is going to move. Just see this wave class. This is nothing but due to movement of one by one cilia, sequential movement of cilia in one direction, right? Now, the ciliary locomotion is coordinated by infraciliary system. We have already discussed infraciliary system consists of kinetosome plus kinetodecimal fibers plus your kinetodecimata. These three are going to form your kinety. All kinety is going to form your infraciliary system. Infraciliary system, right? Now, the infraciliary system is the the ciliary locomotion is coordinated by infraciliary system through the neuromotor center. Who is the neuromotor center? It is your motorium. So infraciliary system along with the motorium is going to form your neuromotor center, right? Ciliary locomotion is faster. During differentiation of cilia and flagella, we have seen flagella locomotion is slow as compared to the cilia, right? Now flagella are responsible for the movement of organisms. Cilia brings out the movement of either of organism or the surrounding fluid as seen in the ciliary mode for the feeding. For example, if I talk about the paramecium, when the cilia is moving, what is happening? The fluid is moving and the food which is present in the fluid will move towards the paramecium, right? Now the last one is your gliding locomotion. Gliding locomotion is nothing but it is the small zigzag movement in the protozoans caused by contraction and relaxation. Simple example if I want to give you. Now suppose if you have eaten something. So here is your mouth. Okay, this is the mouth of किसका बनाते हैं? साकेत का बना देते हैं, ठीक है. This is the mouth of Saket. Now Saket has eaten something, he ate something, and now the food is moving down in a esophagus. Now we all know that how food moves down, obviously gravity to hai hi hai, but what is happening in the esophagus? You will see peristaltic movement like this due to contraction and relaxation. Due to contraction and relaxation. So contraction, relaxing. Contracting, relaxing. Can you see the zigzag kind of movement is happening? Same thing will happen in the gliding locomotion. So class, which structure we have studied for contraction and relaxation? We have studied about the myonemes. Myonemes are the structure which helps in the contraction and relaxation, right? What does myoneme consist of, or what is myoneme? Myoneme is nothing but they are the contractile fiber which is made up of your actin and myosin, right? These are the protein, correct? These are the protein. Myonemes occur in flagellates, we have already seen, then sporozoans, nidosporan, and some ciliates, right? Now, is it correct to say all undulipodia do not show undular movement? Write it in the comment box and answer this class. So that's all we have in today's class. We have studied all the 4 locomotion. Please see this video again and again because it is little difficult to understand. If you're not able to understand, let me know in the class, right? So that's all we have in today's class. Thank you so much.